In this video, we will discuss about antigenicity and immunogenicity. Good morning, welcome to YouTube channel, Bioconcept, Learn with Concept. As we already know that, immunity is a defense system of our body to protect from foreign particle. In this way, antigen is any foreign substance, which induce the production of antibody in our body by interacting with immune cells. On the other word, antigens are substances that react with antibodies. The ability of antigen to induce immune response by generating antibody is called antigenicity, and any substance which have ability to induce B-cell and T-cell mediated immune response in our body is called immunogen. The ability of substance to induce B-cell and T-cell mediated immune response is called immunogenicity. However, all antigens have ability to generate immune response. Immunogens is also called complete antigen. Immunogen or antigen or epitopes that induced immune response by producing antibody or sensitized lymphocyte, which in turn react specifically with immunogens, which produce them. Although, all molecules that have the property of immunogenicity also have the property of antigenicity, there are some properties of antigen is called antigenic determinant. A number of factors have been identified that make a substance immunogen and antigen. Some of the important determinants of antigenicity include 1. Molecular size 2. Foreignness 3. Chemical structural complexity 4. Stability 5. Biological system. If we talk about molecular size of the antigen, then in general, protein molecules with large molecular weight are highly antigenic. Substances with molecular weights of about 100,000 and more are highly immunogenic, while substances with molecular weights of less than 5,000 are generally not immunogenic. And if we talk about a foreignness of an antigen, then a molecule must be recognized as non-self, or foreign particle, to be immunogenic. The molecule is considered self or non-self by the immune system. Then if we talk about the chemical structural complexity, then highly complexity of antigens are more immunogenic in nature. Structural complexity of a protein contributes to its immunogenicity. Chains of single amino acids or single sugars are poorly immunogenic, but if different amino acids or sugars are combined in the same molecule, the immunogenicity is greatly enhanced. In cell-mediated immunity, the response of T cells to the peptide component of the proteins depends on how the peptide is recognized and presented by the MHC cells. Therefore, the structure of protein plays an important role in its immunogenicity, especially in inducing cellular immunity. The lipid-specific antibodies are not easily produced, hence, they do not play a major role in immunity. And if we talk about the stability of antigen, then highly stable and non-degradable substances, e.g., some plastics, metals, or chains of the amino acids, are not immunogenic. This is because internalization, processing, and presentation by antigen-presenting cells, APCs, are always essential to mount an immune response. On the other hand, if a substance is very unstable, it may break up before, and APC can be internalized, and hence become immunogenic. Large, insoluble complexes are more immunogenic than smaller, soluble ones. This is because macrophages find it easier to phagocytose, degrade, and present the insoluble complexes than the soluble complexes. And if we talk about how biological systems are responsible for antigen, then biological system also plays an important role in determining the immunological efficiency of an antigen. Some substances are immunogenic in one individual but not in others. This is due to the fact that individuals may lack or have altered genes that code for the receptors for antigen on B cells and T cells, or they may not have the appropriate genes needed for the APC to present antigen to the helper T cells. The dose of antigen and the route by which it comes into contact with the immune system also influence immunogenicity of the antigen. Very low doses of antigen do not stimulate immune response. Generally, antigens are administered by the parietal route to produce good level of antibodies. The antigens can be given by intravenous, subcutaneous, intradermal, intramuscular, intraperitoneal, and mucosal routes. Usually, the subcutaneous route of administration proves to be better than intravenous routes at eliciting an immune response. Adjuvants are also enhancing immune response. Adjuvants are the substances that, when mixed with an antigen and injected with it, boost the immunogenicity of the antigen. Adjuvants increase both the strength and the duration of immune response.